A very warm good afternoon to everyone present here. Really, we people are very fortunate and we are honored to have Dr. Achyut Adhikari, Associate Professor, Central Department of Chemistry, Tribhuvan University, Nepal, as a speaker for this session. Dr. Adhikari earned his PhD in chemistry from the University of Karachi, Pakistan, and his research focuses on the exploration and structural elucidation of natural products and synthetic compounds, as well as on the study of their phase 1 metabolism. He has completed a postdoctoral fellowship at the University of Sao Paulo, Brazil, and he has actively presented his research in numerous national and international seminars and conferences, contributing to the advancement of knowledge in natural product chemistry and metabolic studies. He has published more than 150 research articles and also patented his research work in the field of medicinal plants. His specific research interests include phytochemistry, the discovery of new bioactive natural products, and green synthesis of nanoparticles and their applications. He is a recipient of the prestigious 2019 Attar-ur-Rahman Award in Chemistry, recognized by UNESCO for his work on isolating compounds in regional Nepali plants for medicinal and commercial products. With his strong background in structure determination and metabolic profiling, Dr. Adhikari serves as an inspiration for young researchers pursuing cutting-edge work at the interface of organic, medicinal, and analytical chemistry. So on behalf of the organizing committee, please join me in extending a very warm welcome to Dr. Achyut Adhikari, and I invite Dr. Adhikari to deliver his lecture. Please, sir.
Thank you, Professor, for nice introduction. Namaskar and good morning to all. It is indeed a great privilege to get a chance to present in front of big scientists, professors, and I would like to thank Indian Chemical Society and Madras Christian College for this opportunity. So today I am going to show something about what we are trying to do. Yes, we don't have that much sophisticated lab, and my presentation will not be like a previous professor's presentation. But what we are trying to do, I would like to just share some of our work here. See, I am from Tribhuvan University, Nepal, the General Secretary of Nepal Chemical Society and Science Director of FACS. You know, nature and medicine. You know, what is the first medicine? Amrit. After the centrifugation of sea, Devata, God, they centrifuged the sea and they got the Amrit. That is the first supernatural product or super medicine. And see, then Sanjeevani Bhuti. Hanumana brought from Himalayas to the Lanka. That Sanjeevani Bhuti is the super medicinal plant. You know, if you go our history, our knowledge of history, so still there are, you know, that previous Professor Rawat has presented some natural product and synthetic compounds as a drug candidate. Still there are around 30% of the drugs are coming from natural resources, and in case of cancer, it is more than 50% are from the natural resources. But you know, only 10 to 15% of the plants from old is explored, and lots of plants are still unexplored in in Asia and in South Asia. See, this is the chart of the this from natural product drug. Still there are many drugs from natural products, and we have just taken the lunch and our blood sugar is increasing, and many of we are taking the diabetes medicine and insulins. So I would like to share some of our work related to diabetes. So, this diabetes is a huge problem. Now in South Asia also diabetes is a huge problem. Like in every house there is one person who is suffering from diabetes. So we try to search some compounds from diabetes from traditionally used medicinal plant. Ayurveda is rich literature for medicinal plant, and that plant which are used in Ayurveda and other different culture like Iranian culture, African culture, they use in the medicinal plant to cure the diabetes. See, we all know that insulin and diabetes, like our pancreas secretes the insulin. If our pancreas does not secrete the insulin, we get one type of diabetes. What insulin do? Insulin is like a doorkeeper. Insulin open the door for sugar to enter inside the cell. After that, inside the cell there will be respiration, and then we get the energy. But if there is no insulin, that glucose sugar cannot go enter inside the cell, and then we don't get the energy, and the sugar will be low, high in our blood. We screened more than 20 traditionally used medicinal plants from different countries to search insulin secretagogues. For insulin secretagogues, out of them mostly these compounds are these plants are used in South Asia like India, Nepal, Bangladesh. See, this is a plant we say Scopia dulcis chinijar. This is also found in India and Nepal also. This plant is also used in the Ayurvedic formulation in India. Gudumar and this plant is also used in the Ayurvedic formulation in India. Some scientists from India has reported that this, Professor Latha from India reported that this extract of this plant is very good for insulin secretion and against diabetes. Then what we did is that we isolated compounds from this plant. First time isolated this compound, very small compound, and this compound was showing insulin secretion, better than this drug, tolbutamide. And in dose-dependent manner, it is 200 micromolar better. And this is the figure of this pancreatic cells, beta cells, that after giving this molecule to the mouse, of diabetic mouse, this pancreas of the mouse is secreting the insulin. Then with collaboration with Bangladesh professor, we have done some in vivo study, because only in vitro study, it is not enough. We should go the in vivo study for validate the in vitro result. And in vivo study also, this compound is showing insulin secretion activity with this some sort of CAMP-mediated signaling pathway mechanisms. This is not toxic. And this, we have done toxicity study also, and this compound is not toxic. This is a very small compound, it is not toxic. And this slide is not my work, but I just want to show this slide. This is post work from again professor from India. After we published that paper, that molecule is very good for insulin secretion. That one research group from India, then they synthesize 35 different derivatives of this molecule. Different derivatives of this molecule, they have done this in silico studies also, and they have done the insulin secretion activities. And they have found that some of the derivatives are 100 times more active than in this coxazole. And this is the beauty of drug discovery in the natural products. Because like artemisinin, that before lunch, Professor Rawat was showing that artemisinin was from the artemisia, but artemether is 100 times more active than artemisinin. It's the same case, like some compounds are more active than standard drug and more active than our molecule. I was interested to do this, but I have limitation of lab and facilities but it is okay. That this is someone else need to do. Then another plant, this is a paper, Nepali paper, we say Timur. Tambul in India, Jantho Timur. We say Timur, its name is the Nepali paper or Siachen paper. This fruits of this plant, we use daily in Nepal with chutney. It has many benefits. It is written in Ayurveda and many benefits. And I started working on it. Then we isolated two flavonoids. Both are good in this insulin secretion. It is not working, I think. And beauty of these compounds are that, you see, if you have no blood sugar, low level of sugar in your blood, this compound does not secrete the insulin. Like now you are taking the lunch, you have high blood sugar. Then these compounds secrete the insulin. If you use these synthetic drugs always secrete the insulin. If you don't need, they secrete. That's why sometimes you need to eat chocolate. So these compounds advantage is that when you don't have high blood sugar, and these compounds doesn't secrete the insulin. See, this is secreting insulin only in the high blood sugar concentration. And we have done some in vivo studies for up to 28 days. Then this is also showing good result. And this is another plant. This is found in northern Kashmir. This plant was actually from Kashmir. See, this plant also used in the northern Kashmir and northern Pakistan, Afghanistan to manage diabetes. People use this plant to manage diabetes. From this plant, we got many molecules, but one molecule, which was major flavonoid, is potent active for insulin secretion. You see, this is also better than the standard drug, and it is also showing insulin secretion in high blood glucose level. So these are the compounds. And another plant, the Salvia mirzaeni, this is Iranian plant. People say name is Iranian, this is Iranian sage. This plant is extremely used in Iranian traditional medicine to manage diabetes, and many Iranian medicine they use this. And we isolated three major flavonoids. Out of three, two are active, and this ASM10 is so nicely so active, double than the standard drug. And these flavonoids are also secreting insulin when this rat has high level blood sugar. And another plant, this plant is from Africa, Sudan. This is the name of this Georgia alata. This name is Gudgand. Good is in South Asia. Good we say for sugar. Good. I think in Africa also they say good or something. But this plant also used by African people to manage diabetes. And there is a paper that Journal of Endocrinology. There is a paper that extract of this plant is lowering the blood sugar, it is secreting the insulin. So, with collaboration with scientists from Sudan, we isolated these compounds, and this compound number 2, very small flavonoid, it is so active for insulin secretion. We should continue this work, but because this plant and this guy is already not in this country due to some problem, then I am here in Nepal, so I could not continue this work. But if some scientists can synthesize this compound number 2, because this is super active, this flavonoid is super active for insulin secretion. So it will be the, and I don't have that much knowledge for organic synthesis. So if we get some collaboration for synthesis of this molecule, then we can have better results. And this tamulin from this Xanthozylum aromatum, this timur. If you read in Ayurveda, timur is hepatoprotective, hepatotonic, liver protective. So then we got this tamulin in a high amount. Then we perform this liver hepatoprotective activity. Then by damaging the liver of the mice by injecting this carbon tetrachloride. You see, I will see these pictures. You see, this is the histopathology of this, healthy rat. No treatment. This is injected. That means this ruptured the liver. Then this is for standard drug, silmyrin treated liver, and that is this tamulin treated liver. Actually, I need to repeat this experiment because it is looking 100% protective. Sometimes if it is more active, we need to repeat this experiment. That's why we have not published this yet, because this is showing 100% protection of liver. And if you read Ayurveda, in Ayurveda, this timur it is written that timur controls the blood pressure. Blood pressure controlling is related to vasorelaxant, relaxing of this artery, muscle of our artery. So, with collaboration with professor from France, we perform this vasorelaxant activity, and this compound was showing very good extract, and compound of this timur was showing very good vasorelaxant activity, and we published this in Phytomedicine Journal. And this is from, I think there was Sri Lankan, I saw Sri Lankan scientists were also there. This molecule is from mushroom from Sri Lanka. Mushroom from Sri Lanka. That mushroom, when I visited Sri Lanka during the conference, and that people were saying that we have an extract of mushroom showing very good anti-cancer activity, but we could not isolate the compound and structure. Then I told them, let me try, because I was working in. Then we got this molecule. There was many molecules, but we could not isolate that. But we could isolate this molecule and we were able to make the structure. And this molecule is very good showing very good anti-cancer activity in nanomolar. Nanomolar in this rhabdomyolymus cancer cell and hepatocancer cell. And this is, this molecule is not attacking normal cancer cell, normal cell, only attacking the cancer cell. It is in nanomolar activity. Yes, this is apoptotic figure that this molecule is killing cancer cells. And then we got one US patent for this compound because this compound was very active in nanomolar. Now we have given this molecule for a company, let's pray that something good result will come. Because this compound, we cannot do beyond this. We just isolate compound and showing that it is very good anti-cancer active and US patent, but we cannot go beyond this. Then we have given a company. Then this plant was also first time this plant, we isolated this compound from Cameroon. But later we know that this plant was also present in the Sri Lanka also. Sri Lanka. And this, we isolated this molecule, and this molecule is cytotoxic for many cancer cell lines. Many cancer cell lines. This molecule is showing this Wnt beta-catenin pathway inhibitor. This is one mechanism of like anti-cancer drug discovery. This Wnt beta-catenin pathway inhibitor. Then there are very few molecules are there for this inhibitors. And this molecule is very, I think it is not showing there nicely. And this in the molecular docking and I am not going to, I think, where is this? And if you see the figure, this molecule is like destroying the cancer cells. Then this ICP value better than paclitaxel, standard drug, Taxol. So this molecule could be a lead molecule for another study, and this molecule is showing this apoptosis and many proteins is caspase activity. That means this molecule is destroying the cells. That caspase enzymes are releasing there. And if you see there, this molecule is suppressing the cancer causing gene and enhancing the anti-cancer protein. This is the beauty of this molecule, that this molecule is suppressing the cancer causing genes, and like this is enhancing the P53, and but suppressing these are cancer causing genes. This molecule is suppressing cancer causing genes. And if you see this molecule is enhancing the P53 protein, which kills the cancer cells. So this molecule is has a potential to study for further. Then another molecule, we see Kalanji. This set, this Kalanji is we use every time. And it is, if you see that this Kalanji is even anti-cancer, it is written many things. So we started working this Kalanji. Then this then we got this saponin, and this saponin from Kalanji is also very good for this Wnt beta-catenin pathway inhibitors for anti-cancer activity. See, there are lots of simulations and in vivo. And if you see there, this molecule is also this molecule has also better IC50 than paclitaxel. Paclitaxel has 64, but this molecule has only 24 micromolar. And if you see, there are different types of this molecular level separation, that this molecule is also suppressing the cancer causing genes and enhancing the anti-cancer molecule. See, this molecule is suppressing cancer-causing gene and enhancing the anti-cancer protein. So this molecule from Kalanji, maybe there are other molecules in Kalanji, because our literature has written that Kalanji has many importance. That's why we always take Kalanji. So and anti-filariasis, we see hatti paile, something people have a big foot, and this is a parasitic disease. This is also a molecule from Sri Lanka. I am visiting professor in Sri Lanka, this IBMBB Colombo, and I supervise student there, co-supervise. And when I visited there, they told that we have a plant extract, which is good for this against the worms of this filariasis. But again, we could not isolate the molecule. Then I told them, they go, okay, let's try. Then this extract, different extract are for larvae and even the worms of this filariasis. And it is actually, we got this small structure, active one. We got many structure, but this small structure from this actually turmeric, wild turmeric. ये जंगली हल्दी है, जंगली हल्दी. This is जंगली हल्दी. People use this, that paste of this जंगली हल्दी in their food when they have elephantiasis in Sri Lanka. I think Jaffna or somewhere in the Sri Lanka they use this जंगली हल्दी. And this, if you see this molecule is like neutralizing these worms and larvae of the filariasis. And this is another molecule, active molecule from Nepali plant near to Himalayas. In Tibetan system of medicine, it is written that this plant is can be used in anti-cancer. This Cordialis gobineana. Then we got a large number of like huge compound from this plant. It has measured, and it is active against the breast cancer cell. And in against leishmaniasis also, I did this with collaboration with Brazil. When I was doing my postdoc, this is my postdoc work, dearly. Still I am showing my postdoc work. In other bioactivities, we have done lots of bioactivities of this molecule and published many papers. It is in hepatoprotective and many other things. And this plant, this is unfinished work. This is a plant we say Mahonia nepalensis. This is endemic plant in Nepal. Major part of this plant is berberine, but we got from leaf this molecule, and this compound is very good active for anti-inflammatory. Then why I am interested in this molecule is that this molecule structure is very close to the drug silmyrene. Only one position different there, here. Silmyrene has OH and it has CH3. Only one position is different than this. This silmyrene also natural product. This molecule has a very close relation. So it will be our future project. And essential oils. Essential oils are nowadays people are using Sugandhit essential oil, aromatherapies. It is very popular. So I tried to do in collaboration with South Asian University, Delhi, because in my lab, in my university in Nepal, we don't have that much sophisticated anti-cancer lab. So I do this of cancer with collaboration. And this essential oil of this again this Timur, Xanthosarnum armatum, is very good against this lung cancer cell line. If you see the figure, you know, this in 200 micromolar per meter, there is a complete cell, you know, destroyed. This essential oil of this Timur destroy the almost complete lung cancer culture. In the culture plate, this in 200 micromolar per microgram per mole, per ml, this almost all the. If you see this, you know, if you this anti-proliferation, that means this oil stop this, you know, proliferation of the cell. Because if you see here, that there is a growth of cell, that there is a space. But when you inject this food plate, there is a space. That means this oil, the essential oil, stop proliferating the cancer cells. So this is also an advantage of this work. And you know, if you see there, this oil is also banishing some cancer genes and enhancing the you know, beneficial proteins. Then there is another. How time I have? 2 minutes. You know, there is another plant, and in Nepal, people use, if you have sinusitis pain, people were making powder of this leaf and they were smoking. They were smoking and they will get relief of this pain. So I thought maybe there are some volatile molecule and it goes, some molecule must be there and it is working anti-inflammatory. So then this essential oil of this compound, so yeah, it has myrcene and beta-ocimine and linalool as a measure. And this, if you see that, you know, anti-inflammatory activity, this essential oil is showing a very good anti-inflammatory activity. This is suppressing TNF-alpha, COX-2, beta-IUNOS. You know, it's traditional knowledge. Science is supporting the traditional knowledge. You know, our ancient, they had their science, their level of science. They were trying with different plants and interestingly, they were smoking this you know, powder of this plant. They were smoking, and actually smoking is not good, considering good of health. But essential oil of this plant is beneficial for long time. So they were smoking this. Okay. And there are some different slides. I don't have a time, but because, you know, these are some work where there was a COVID and labs were closed. We have to do something. Then we start doing this in silico work. In silico work and sort some natural products and isolate again and do activities. And such type of works are there. They are also all natural products. And against dengue. Okay. Acknowledgement. I would like to thank Madras Christian College and Chennai, India, and Indian Chemical Society, SRM University, Professor Pokhrel from South Asia University, then my postdoc supervisors, my collaborators, and Professor Samira from Sri Lanka, and from the work done in Bangladesh, also the in vivo study. So these are my students. We are only reaching the young students. You know, we have a number of young students, bright students. We don't have sophisticated instrument, but we have young students that they do something in the desert also. They get something in the desert also. So, this is Nepal, Pashupatinath, Pokhara, and I will invite all of you to visit Pashupatinath. This is once in your life. You should come. We have Muktinath also, but Muktinath is a little bit far, but Pashupatinath is very near to airport, only 1 kilometer. And if you can see lots of beautiful mountains. So, thank you.
Thank you, sir. Thank you for your interesting talk. Since we are running out of time, at least any one question from the audience. If you have any clarifications or queries. So, in the absence of question, let us conclude. Really, it's very interesting, sir. We know the difficulty in starting from the plant collection and then extraction, isolation, and then identifying the bioactive compounds from the isolated molecules, and then to evaluate all the biological activities, starting from diabetes to your anti-cancer activity. Really, it's wonderful. Thank you, sir. Please join with me in congratulating our speaker for his wonderful talk. On my own behalf and on behalf of the organizing committee, I thank once again the speaker, and I would like to honor the speaker. Thank you for this opportunity. Thank you.
Thank you, Sonalata ma'am, for chairing the session, and thank you, Achut Adhikari sir, for your insightful presentation. The next one is we will be having the industrial conclave and the panel discussion. Can I have the media team to project it? Yeah. We will wait for the panel members to be here. It will take quite some time.